I'm showing you here an example of, again, that same image I showed you at the beginning of the talk, showing the white matter lesions. And on the right is an example of something that comes from perfusion-weighted imaging. And in this particular case, all these red spots here, you'll notice match all the white spots over here. And in this particular experiment, what we did is we took the information from a single region in the 3D volume of your brain, and we asked what parts of the brain behave exactly the same way as that MS lesion itself. And if there is a vascular connectivity associated with this, all the lesions should turn red like this. And you can see they do. Actually, it's quite amazing. The fact that I only chose one lesion at one slice actually highlights all the lesions in the brain. It's another indication that there is a vascular problem associated with those lesions, because they all behave the same way. Now, you can also use MR perfusion to study what happens in acute lesions, and it's well known that in acute lesions, CDV goes up. So the body's first response when something like this happens is to increase the local blood flow. And although it may be hard to see, this little red circle that's here is also here, and the green here means increased blood flow. So that's what happens on an acute lesion. But on a chronic lesion, something that's been around for a long time, like this one, if you look here at the cerebral blood volume, there's basically nothing left. If there's no blood getting into that tissue will become ischemic, eventually necrotic, and it won't function anymore. If it has no blood supply, it's not going to function. So clearly you don't want lesions to be around for too long. You don't want them to get chronic. And here's this perfusion example I was alluding to. If we look at these medullary veins that can be seen very nicely in a normal person, in someone with relapsing remitting MS, you can still see them, but maybe not quite as clearly. Someone with more progressive MS, it's hard to find them. So the question is, what's happening here? We should have seen them better, like in the caffeine example. But what is probably happening is that there is some contraction of these veins, or some reduction in the local cerebral blood volume. And of course, if I reduce the blood volume, I will see this effect much less, just like we're seeing here. And that all, again, matches the concept of reduced perfusion, reduced CBD for MS treatment. Okay, another very busy slide, so I'm going to skip this one. You can read more about ischemia. Ischemia is related to low perfusion. It means the poor delivery of oxygen to the tissue. And it's been shown, actually, by Bill Bradley, probably about 20-25 years ago, that if you have an ischemic event, even if you're a child and you have a serious ischemic or anoxic hypoxic event, that you will have an increase in iron content following this. And one of the hypotheses of that is that if you disrupt the iron transport that would normally take place, say, out of the basal ganglia or the deep gray matter, then that may lead to iron increases, similar to what we see in MS. And they actually demonstrated that that same type of reduced perfusion, ischemia, but as a severe acute event, led to almost immediate iron deposition. So, in fact, it's very similar, perhaps, to what might be happening to MS. And again, it's painting a very coherent picture of what the role of vasculature is. So, another interesting point here is the work of Pat et al. They did some studies on gerbils, and when gerbils were fed a very low iron diet for 8 weeks, they had decreased brain and serum iron levels, and they had less neurological deficits and decreased brain edema after temporary unilateral carotid ligation, kind of something like what you might expect to happen if you have a stroke, and then reperfusion than the animals who had high iron levels. Now we have to be careful here, because iron in the blood is not the same as iron in the brain. So you don't want to go out and not be taking iron. It's how that iron manifests itself. Iron can appear in the form of ferritin in normal tissue, and if you bleed, it appears in the form of hemosiderin. So there are different types of iron associated with this. I think the critical issue here is also shown in Grant et al. That mice that are usually used for this other model, the EAE model, the experimental autoimmune encephalomyelitis model that the neurologists use a lot, it's not the same as the MS that you have, but it's similar to it. That these animals don't develop EAE if they're on a low iron diet. So a totally different study, but similar types of conclusions. So there are some interesting things and studies to be done with iron. So we can use what we call phase images here to map out iron in the brain. So here we have iron in the motor cortex, shown very clearly with this type of image. You can see nice contrast between white matter and gray matter. Using that same technology, we can also find that there's iron in MS lesions. And here you can see a Dawson finger with iron in it. Here you can see actually the connecting draining vein that first comes through this lesion from here and then drains out to the edge of the putamen and follows here. So just as you may have read in the past that many of these lesions are venocentric, you can see that quite clearly in these images. But not all lesions have iron in them. According to Adams, who did some cadaver brain studies in the 1980s, only about 20 to 30% of lesions have iron in them. And some people may have lesions that have no iron at all. Others may show, as in this example, iron, again, another Dawson finger example, iron here, lesions in here and here. Here is an individual that shows iron at this point more distributed and yet iron in a ring-like structure. And here's a third individual whose all the lesions look like ring-like structures. And so these are called type 2 and type 3 MS lesions. And they can be very different from one MS patient to another. So clearly there's something different going on there. And how these things develop, we don't understand yet, but it's very important for us to realize that the response of each individual person to a different type of vascular challenge may be very different. And Zamboni, as well as Adams, stained the small veins called the venules to see if they had iron in them. And sure enough, that in some of these cases they found iron inside the vessel wall. That's not a healthy thing, that's not a normal thing. So those images that we're looking at do demonstrate this behavior. Here we have an example of a normal young person. You can see the thalamostriate veins, similar to what I showed you at the beginning. There's no hint of iron here. So again, to quote Burton Dreyer from Mount Sinai, as we get older, we all rust. We all have a certain amount of iron that increases over time, but at 20 and 30, there's not a lot of iron. This is an MS case. And here you can see that there's iron at the confluence of the draining veins in the caudate, lobus pallidus, and the putamen. If we image him a year and a half later, this iron gets worse. So we do have some demonstration that the iron buildup is following some veins pathway. We've also shown that iron builds up in the pulvinar thalamus of about half of the MS patients. So we have a lot more evidence today that iron is related to some of these vascular problems. So I'm going to skip a little bit here and just show you this image, this graph, that plots iron in 100 normal people. And you can see that the pink and the blue here, they increase in their iron content as we get older, from 20 to 70 years old. And the X's and triangles represent MS patients. And you can see that the young ones here actually have very high iron content associated with them. And many of the older ones, not so high iron content. That may be because we're looking at two different groups of people. Very rapidly developing, young people who have rapidly developing MS, and they may not appear on this chart because they may not be around after 40 years old. If that were the case, we might see the iron continue to go up and up like this.